Hello, everyone.
Hi.
Hi. Hello. Sriram Muthukumar. I go by Shri, and I'm a CEO and a co-founder of EnLiSense. With me, I have Vikram Dhamu, who is my systems engineer and operations manager. And then my other co-founder, who's not here, is Shalini Prasad. She is also a faculty and head of department of bioengineering at UT Dallas. So today I'll be talking about how EnLiSense got started and what we do. I'll go into the details of it, but it will be about our sweat sensing technology, which is a wearable platform that we have. And it's pretty innovative. We developed it. We have been working on it since 2014, and we're working towards getting an FDA approval. So I'll be sharing about it. It'll be different than IoT, but that's what Ed wanted me to introduce, and how you're using sensors to the whole connected world.
You got a lot of sweat equity in there, huh?
Yes, literally true. So since 2014 is when we started. It started out of a dinner conversation that I had with my co-founder, Shalini Prasad. So I suffer from GERD. GERD is acid reflux, and it has various triggers. It could be not related to food, it could be related to environment and so on. And her mother also suffers from it. And we were just talking and discussing about how it would be good to develop a sensor that can actually monitor different health conditions. My background is a material scientist. I specialize in solid state physics, and then I spent my 20 years in semiconductor industry, working at Intel, Maxim, Volvo, and so on. So my background is all about solid state science. Shalini's background was biosensing, characterization. So we're talking about it, and she said we could make a sensor that is can amplify the signal over noise, make it robust, make it non-invasive, because I don't like my body pierced for blood sampling. So that's how the discussion started. And by the way, audible. And so my knowledge in material science combined with her background in biosensing characterization is what we combine. And we developed the stack, which is what we have as a sweat sensor. It's a wearable sensor. I'll pass it around. And then it meets with a device that is size of my Apple Watch, that I can wear it on my hand and monitor it. So that is what the sensor that you see out here, which is this orange strip. It's the size of a thumbnail. And what it does is it passively measures the sweat in the skin. And through that sweat, we look at different biomarkers. And through the biomarkers, we actually go and report the disease state in the person. So we were funded, we started organically. We started to, yes?
Rule of order. Are you taking questions through this, or do you want the questions after the presentation?
It's a free form. So when I spoke to him, it's all up to.
Whatever you want to do.
I'm comfortable either way.
Yeah,
So,
questions begin.
Okay.
All right.
So when I spoke to Ed, he said the group is technology management. So I put some biz dev slide. I put some technical slides. If the questions are more technical, I'll steer to the technical. The questions are more business development, I'll steer to the business development.
Okay, I'll be first. What is IBD for biomarkers?
IBD is inflammatory bowel disease. So it's the one that affects where you get colon cancer due to Crohn's disease or ulcerative colitis. It affects the part of the colon that can get inflamed. So the colon, if you look at it, it's like a sausage where the inside is like made of a mucous membrane. And for either genetic reasons, due to stress or environmental, or there's a lot of factors, people don't really know what the exact causation is. But if that intestinal lining inside your thing gets thinner, then it actually breaks open, and it can create issues, because as the food passes through your colon, it can then merge and create inflammation.
Otherwise known as bad
Yeah. But it's not curable. There is no cure for it. So once you're diagnosed with it, you have to live with that disease. And that became our pivot of how I wanted to go. Our platform is universal. It can measure different inflammatory markers. So how did the story of inflammation came? We first started, as I said, GERD is an acid reflux. We started looking at, and this was back in 2014, we started looking at biosensing. We started looking at markers like CRP, C-reactive protein, which is produced by the liver. So today, if you get COVID, if you get sepsis, if you have heart disease, your liver will send like scouts telling that your body is under distress. And one of the scouts is C-reactive protein. So today, C-reactive protein is measured through blood. And so we started looking at, can we measure through sweat? Can we measure through other body fluids? And so that's how the question started. So we have a lot of publications on different markers. Why IBD? So we were funded by Crohn's and Colitis Foundation. They have invested in my company to develop this product. They want me to develop a wearable product that can be what continuous glucose monitors like Abbott Freestyle Libre, Dexcom is for diabetes.
Ready?
They want a wearable product. Yes, exactly. They want I would want to get to that size. Today I'm at the size that I'm doing. But they want me to make a wearable product for IT. That's my pitch. We were also funded by BARDA. BARDA is the Biomedical Advanced Research Development Authority. It's part of Human and Health Services. They are the ones who developed the COVID vaccine, but they funded the vaccine development and stuff. So BARDA's interest in us was, if you can measure inflammation, can you measure inflammatory markers for sepsis? Because eventually, anybody who gets acute inflammation will develop sepsis if you don't get treated, and you can die from sepsis. So there is sepsis, but sepsis has different cause. It can be viral, it can be bacterial, it can be fungal. So they wanted to, so what we track is we report the markers that are common for what is looked at. Like if you were to go give a blood sample, what are the markers we look for? We can provide the same markers for wearable platform. So they funded us, and NIH funded us also. So through that, all this funding is how we developed and what you see today. And my next goal or next phase here is to take this into a product that I can get FDA, and then we want to go after multiple other disease states, like rheumatoid arthritis, liver issues, and so on. Okay. So that's about me, and we have several IP in publications. Okay, how the name EnLiSense came about, enabling life with sensors. That's what your license. Yeah, go ahead.
I did have a question, sorry, on the previous point. If you go after this different diseases, is it like a different strip?
Yes.
Okay, so basically, if I want to check like a liver problem, it will be a different strip than if I want to check this IBD problem.
Yes, but it will. So the way it works is it's a three-pronged system, and I'll go through that in one of the slides. There is a sensor which samples the sweat, and then that's recorded as a raw data. Raw data gets processed in our app and through the server into a measurable reporting concentration, like how your CGM does for the blood glucose. So the CGM is actually measuring your intestinal glucose. It's not measuring your true blood glucose. But algorithmically, what Abbott has done is it has correlated to your plasma glucose. And when you see it in your app, when you see it as equivalent blood glucose, it's basically converted number. So we do the similar conversion. The algorithm is different, but the process and the logic is the same. But we go after inflammatory markers. So when you look at inflammatory markers, they have a common etiology in the sense that there are cytokines, chemokines, and inflammatory proteins. Like inflammatory proteins can be produced in the liver. It can be due to liver cirrhosis. It can be due to non-alcoholic fatty liver disease. It can be due to NASH. So what we do is we build thresholds based on disease states. And the thresholding is what we tell based on your baseline condition. So today, the CGM that Mike just showed wearing, that is baseline based on Mike's level. It is not based on my level or somebody else's level. And that's what the algorithm adjusts. So when he drinks a sugary drink, it goes up.
I don't do that.
It's relative to that level, right? And And so it's like, think of it as driving a car in a highway on a lane. And if you go to the left or to the right, it warns you and says, oh, you're swearing too much to the left or right. That is the decision support that it provides. And so while it's not a replacement, it's not a diagnostic that replaces a standard of care at a blood test, it's a warning system that can be used for remote patient monitoring to say when the patient has to do the blood test or when the patient needs to be checked into the doctor. So that is what we enable. So that is what this whole power sensor connects with the whole IoT to add value to the patient and to the clinician.
Let me ask you this question. You measure mostly on inflammation protocols. What about other diagnostic, capabilities like pulse ox, blood pressure?
So that's a good question. So that's a segue to what is a biosensor, right? So a biosensor can be of different types. If you have a fluorescence, optical, piezoelectric. So pulse ox is a optical biosensor that looks at what the thing is. An heart rate that your Apple Watch measures is also an optical biosensor. Like if you look underneath this Apple Watch. Then there's piezoelectric, like what Corvo is trying to do, where they look at surface acoustic wave devices or bulk acoustic wave devices to look at sensing. Fluorescence is a very common thing, which is what is used for most laboratory analysis. We specialize in electrochemical. Your glucose sensor is electrochemical. So what is electrochemical is that we can apply these methods, or apply a voltage, measure a signal, apply a current, measure the voltage or measure the signal. We can also measure impedance. We can measure potential. We specialize in non-Faradic impedometric. Why non-Faradic impedometric? It's very much like we measure impedance. There is a real and then there is an imaginary. Within the imaginary part is your, I can go technical, right?
Yeah. Bring it up. Everybody gets it.
Within the imaginary section is where it can be a contribution of a capacitive or a contribution of an inductive behavior. So when an analyte binds to a receptor, there is a dielectric potential that is created at the interface, and that dielectric potential causes a change in the capacitance, and that capacitance is what we measure. So today, when the sweat expresses, and sweat has a lot of biomarkers, right? And so those biomarkers, when they come in contact to the sensing interface, if there is a biomarker of interest, it will tag. It's like a Velcro. It will bind. When it binds, it changes the dielectric value. That is measured as a change in capacitance, which is what, that's why we call it label-free.
Okay, so I get what you do. And there are other types of sensors that are available. From a strictly go-to-market perspective, would there not be value to actually in the same physical case merge the different technologies from the perspective?
So the challenge here is what is measured electrochemically will have to be based on, it can always be corroborated, right? For example, today, your heart rate is based on the optical. Correct. No, but it is correlated to an EEG. So there's always a calibration that is done against a reference. If you ask me today, your blood pressure, what is a true blood pressure? Nobody knows.
Yeah.
It's always against a reference.
And also your arterial and your venous.
Correct. So when you try to mux multiple different methods, you are challenged by the FDA to validate each of the variations from that method. So if your total variation square is a sum of the independent variation square, now you have to do clinical studies with each modality to de-risk it to say the sum total that you provide is the accurate value. So when Abbott comes out with their freestyle Libre, they take it to FDA, they say, I'm measuring glucose, I'm correlating it to blood glucose. That is all I need to show you. I'm going to spend so many $1 million to run the clinical trial, get the validation, get the device out. If they were to now mux it with, oh, let me go do an optical thing, which is what Apple is trying to do. Apple is trying to get an optical glucose sensor. If the optical thing has variation based on the skin color or the skin tone or the access to the blood vessels, and that causes a huge variation, FDA says you have to lower that variation before I allow you to sell as a medical. There's a business, there is a challenge. You don't want to do all in one. Today also, why did I pick IBD? Because when I said, oh, my platform can do any marker, they said, how many markers are you going to prove? So this is where I have to pick and choose. I have to pick a use case, I have to pick a disease, and I'm focusing on that. Yes, my platform can do a lot, but when I have to go to market, I have to pick one use case.
Well, the reason I asked the question from a service delivery perspective, we're actually trying to deliver healthcare. And, you have sensor models, valid, available, in essence, I can, in my mind's eye, I'm envisioning someone's arm with 16 watches on it. Okay, because I'm measuring this, and they're all FDA approved, and they're all individual single source, and that's hugely inefficient for the patient, care operators.
Yes, absolutely.
You have bigger problems than that if you're having to wear 16 watches. You probably need to be in bed at home or in the hospital, but I get your point. It's a good point.
Which is why we pick and choose. So if I were to,
You can start with one and then move on with the other ones as you go, right?
That's true.
But you need to get your product to market first.
that's right. So for me, the first product is the most challenging. Once I do that, that is itself is a predicate for me for subsequent product.
Exactly.
That's how my plan is. It's a platform technology.
So the sensor that you said that you passed around is targeted which is IL-6?
I have a few markers. I'll show you. It's basically disease specific, and there's a reason why we do that. Is we don't. I'm a engineer or a material scientist, so I don't know which mark. So I rely on my clinicians to guide me. What are the markers of interest for your use case? Like for IBD, today the marker of interest is calprotectin. Because today the one that people rely on is a fecal test. Like you have to take a stool sample and you have to put it on a point of care device and they measure the calprotectin, which is a indication of your intestinal inflammation in your colon. So the use case here is I don't like measuring stool samples. It is nasty, dirty, so on. But you talk to an IBD person, they say, I'm having blood in my stools. I'm having constant diarrhea. Today, I can't attend a meeting. I can't function. So I'm looking for the nearest bathroom for me. I wish I had a wearable device that could warn me, saying if I have a pineapple, is it going to give me a flare? If I eat something, it's going to give me a flare. Is my stress going to give me a flare, and so on, right? So then
I get it, and but you're going after these particular ones, and what you're talking earlier about CRPs as a group is kind of a general statement, but you're really narrowing it even further. So
totally, CRP is a useless marker for me. Why? Because it is a sensitive marker that goes on for whether you have a heart disease, whether you have a liver cirrhosis, or whether you have IBD. CRP goes up. It is non-classification marker, but it is today the standard of care. Like if you go to a gastroenterologist, they make you go to a CRP test. But really the marker that is specific for the disease is calprotectin. So when I do it, so here is my strategy. Yes, how many markers would you want me to put? So if I have to do strategic to make sure that both the clinicians will buy it and the patient will endorse it, I have to put the two markers that is today the gold standard, which is calprotectin and CRP. Putting it all on one platform, which is what I can do, and measure it and make it as a wearable that can report and correlate it to what you get with your fecal testing. So you don't need to do fecal testing, don't need to do blood testing. This will be, it's not like getting rid of it. This is like your decision support to say when you want to do it, not randomly, not ad hoc. When you are likely to get a flare, that's when you want to do it.
How long do these last for, and how long do you wear that until you need to get a flare? I mean, you got to change it out every week.
That is correct.
Every shower you take.
That is correct. Actually, you guys are asking, let me. So, as I said, it's a 3 sensor system. The sensor can measure 4 markers at a time. You see each one, there are a pair of electrodes, there are 4 pairs of electrodes. Each electrode pair is one sensor. Now, I can make it a duplex by making 2 markers on both sides and do like a duplicate, or I can make it a single marker by putting all 4 sensors with 1 marker. But at any given time, I can do up to 4.
So this IVD, is that something that, I mean, once you sense that you have it for life, you said, so it's, or something like that?
Yes.
Or
When you go with line, you fails or something.
I need it for life.
and so I'm trying to detect the difference between measuring blood glucose, which is up and down all day. And so measuring that continuously is important versus a marker that says, you put the watch on once and it says within 24 hours, it says you're screwed for life.
No, it doesn't work like that. The way, okay, you asked me about IBD. So IBD has 2, so it's Crohn's and ulcerative. Today, it's not like diabetes. Diabetes has around 30 or 40 million people in US impacted. It's only 3 million, but there is no cure for it. I'm using this primarily to get my first FDA. But still the impact here is pretty high because it's a significant cost burden, right?
So if I don't have the watch, I've got to go do these other tests, which are invasive. But once I have a problem and I go to the doctor, I get tested. I'm trying to differentiate. If I have a problem and I go to the doctor and they do a blood test, I don't mind. What I don't want to do is a blood test every single day.
Yeah.
So I do this
Yes.
as a diabetic. I don't prick my fingers.
Yes.
So that's the advantage of the watch or the semi-invasive.
Correct. That's what we are shooting for.
Okay.
Too. Except it will be exactly the continuous monitoring. In fact, it's not a stock photo. She's our first IBD subject who volunteered for our studies, and this was about 2.5 years back. She was a UTD subject who came in, looked, reading our papers, said, "I want to test it," and she's wearing the device in her arm. This is a typical calprotectin that you see when you're starting to get a flare. You can see it go up. Okay, that's what my thing measures. And what we do is we provide this both to the patient and to the thing. Right now, where I'm trying to do is looking at different N numbers to see what is the alert level. I see it go up. Like your question, is it still in the green zone? Is it in the orange zone? Or is it in the red zone? That's what your Abbott Libre CGM tells today. So today, between 80 to 140 is considered. So how did the CGM decide 80 to 140 is your green zone? Based on statistics, based on race, based on age, based on gender, based on different styles. They came and said, oh, taking a big N sampling, that's what is your average glucose. Some people can have elevated glucose and still be okay, right? So that's what we are trying to work. But this is how it reports and it tracks. The data is shared with the clinician. The clinician can look at it remotely and say, hey, I need you to come in. Rather than you coming in every 3 months, you come in only when the flare-up or likelihood of flare comes.
Cool.
It's the triggers are diet-based, right?
Not
So-
necessarily.
Not necessarily. So it's not tracking like, hey, if I eat a pineapple, like you said earlier, if I eat a pineapple, it might not be.
It's
Producing
usually,
triggers.
it's usually if you have a lot of stress, and then you drink the pineapple. That's what happens.
So both are right, but nobody knows. I'll tell you, this is a funny thing. I work with one of the leading expert gastroenterologists in the US. I'll come to your question. And I work with a group in Chicago, and we actually have done many studies there. The person in Chicago is the leading expert. And I also work with Mount Sinai, which is also a leading center for IBD. Both people have dichotomous, inputs on what causes IBD. And both clinicians, they don't want to say food, diet, environment, stress are the causation of IBD. Why? They're worried the patient will not come to them. They want to say, I have a cure for you, medication is only cure. This is where the whole health cycle becomes where, pharma drives who you go to, insurance, care, and so on. But you talk to, I spoke to this Jewish person who is from an Hasidic community, and the Hasidic community is a very close community. So they have the incidence of Crohn's disease. People don't talk about it. You don't want to talk about poop or blood in your stools or diarrhea, but there is a thing. So what is the triggers for one community may be different for another community.
I get gout. It kind of reminds me of gout. Like sometimes spaghetti sauce, like red wine. That's kind of what I remember. It's gout.
Sorry, there was one more question.
Are you able to track the thresholds or generalizing the thresholds for standard population?
So this is the data for 24 hours. So this is a person who is healthy, has no IBD, no situation. You can see the levels, the blue line. The red line shows the person is from ulcerative. The person was reporting intermittent flares during the period. You can see how it goes up. There could be a circadian pattern to it. You can see that the day-night thing. We don't know, but that's the N number is too small. But yes, I hope this answers your question.
But is it relevant for all kind of communities?
No, I'm collecting the data because even then, if you look at Crohn's, ulcerative colitis, right? The levels for active is different than Crohn's. They both disease fall under IBD. But when you are in remission, they both are at the same level. So when you're flaring, your flares can be a mild flare, medium flare, severe flare. So that range is very wide. So that, but then when you're coming back to remission, because the whole cycle here is wellness to illness, but back to wellness, right? If you are in a disease or a distress state, the goal here is to get back to the healthy state. So the drive to go back from illness to wellness is the character. Today, the CGM that Mike is wearing, if it triggers an alarm, what does he do? He goes and tries to drink more water.
Yeah.
Or because the whole idea is to get back to the wellness state.
Watch does that well.
Yes.
I'll tell you, this is the most awesome thing ever made for diabetics. It's cheap. It's $35 retail, 2 weeks. Even if your insurance doesn't cover, $35 Walmart every 2 weeks. Everybody can afford that. It's cheaper than the
Does it
insulin.
integrate with an insulin pump?
Oh, you can integrate. This does not. You buy the full CGM, but those are very expensive. But for type 2 diabetics or insulin dependent, or type 2 that are not insulin dependent, those are both can't get the pump no matter what, or the CGM, and this works great with that. And then the super benefit is just you can constantly monitor your health. So you go eat something, you go, that didn't go well. You know immediately. So I know you're not pricking your finger 20 times a day. Don't eat that.
There are false positives, but the key thing here is it alerts you to go take the necessary proactive action.
It's a game.
Yes.
I've turned it into a game.
That's what habit is. I need to.
So, and my background is with Draeger, with drug and alcohol testing. So your charge state of the analyte determines the bond with the electrochemical device, with the device. So as time progresses, does it become less sensitive because the electrochemical bond is have so much?
Good question. So the one I sent to you. So today, the way I had to engineer the thing, I don't know if you saw the script that I sent, right? So the size is a thumbnail, right? The one I showed you.
Oh, yeah.
So today I used sedentary sweating rate of 1 nanoliter per minute per gland, right? And so if you look at the area per 10 millimeter square, what is the gland density times the sampling rate is my sweating rate every minute, right? To that, over time, it will get used up. It's an accumulative sensor.
Okay.
So that's why it lasts about, I think I showed, it lasts about 4 days.
But does the sensitivity then decrease as it's used?
That's what I optimize for. So it's like your half-life.
Okay.
I
yeah.
stop it at that and go longer, but that's when I start to see it. So there is a person CV. Once the person CV goes beyond that, you say, okay, time to replace the sensor. Yeah, right.
So is the capability of integration of the viability of the sensor a function of like how much you sweat? So if you're like living in New Orleans versus living, I don't know.
So today we ran this test. So these are all the studies that we did. So this use Chicago Medicine We ran it up about 18 months across the different, like summer to winter. Here we had like 20 active, 20 inactive. We did the Mount Sinai. We also did one with the Veterans Administration in Virginia, where we looked at liver cirrhosis. We are doing it in Dublin, which is very close, because there people of Irish origin, I'm told, have more incidence of non-alcoholic fatty liver disease. So that's where we're looking at NAFLD NASH subjects with dietary intervention. It's actually very cold and dry there. And so, this year I'm doing one site in Europe, where Greece, Athens, which is more Mediterranean. I'm collecting data. So far, we don't see the issue, and we are trying to normalize it. But it will be in my sensor. So right here, I have my skin temperature. I also measure my perspiration, then the 4 markers that I'm monitoring. So I use my perspiration and my skin temperature to guide me to say whether I'm in the, because at the end of the day, it's the local sampling that I'm doing.
Right. Do you know when the sensor has sort of that it's useful, like green skin? In other words, let's say the person went running a bunch of times of like a gig, right?
So that is a very false notion that people think, and this is the problem where you get papers from big schools like Stanford and this California school, but they don't understand one thing. I'm sorry to answer it that way, but no ill intention. I tell you why. Sweat means it's also trying to cool your body. If I go run on a treadmill, or you go stand outside in 100 degrees weather and you come back and you start to sweat, you're just producing more water. We are not measuring the water. We are measuring the marker in that sweat. The marker concentration has not changed just because you went out and came in, or whether you ran on a treadmill.
Right, it was my question about the viability of the
So
system,
the durability, the sensor gets,
So that's what I've done over the clinical studies. It's still the 4 days lifetime.
but you sweat a lot, for instance, the adhesion might lose. There's other environmental factors that impact the sensor.
Yeah, but not doesn't count. Yeah.
But it seems to,
You just
So
get
far.
better glue.
No, so far. I mean, am I going to fail? Maybe, but that's what my N number is, to go catch more data. I mean, engineering mentality. 1 minute. Engineering tells me that something will fail at some point of time. And that's why my intent of running so much studies, and why I've been at it for 7 years to get all of this data. So my platform, yes, I always am looking for room for improvement. I've not had the kind of issues that you're asking. We have not seen it. Yes, go ahead.
Your data gathering is admirable. And might be a good case study to follow some folks in Japan and do case studies there because of diet, other things.
So the study that I'm starting in this fall, I'm actually going, is in Greece and Prague and Zagreb. That itself is a challenge for me because one, I'm doing it with kids, 7 to 18, and I have to deal with the language conversion. And my platform is right now on Apple, which is the iOS platform. So I'm going to test this out, and I'm going to see how this works, because I have to now communicate what I've been telling in English in their local language. Then I'll go work with other
You're going to
countries.
track the Mediterranean diet.
There is a dietary intervention, yes. So the way the study is done is, these are people who are diagnosed with Crohn's disease, who are actually coming after a dietary cycle, and they're going to be going on a liquid diet. So over a month, we're going to collect their stool sample, and we're going to look at our sweat sensor data, which is what you're asking, right? How am I building correlation? Yeah. Is I'm going to correlate to the calprotectin, what you see in your feces, what you see in your sweat. Are they related?
This is like a bunch of shit.
Yeah.
Do you keep it on in the shower? Or
Yes, so actually the way she's wearing it, I use a adhesive patch on top of it. Initially, some of the CGM people also wear it. So I use the same FDA approved patch to hold it together. And so that is waterproof. It is not, I've not tested it for like a swimming or a diving type of,
It
like if you go to, it doesn't work, right? But
It works great for shower.
it's great for shower.
Yeah.
It's made by 3M. And that's what I use. That's the setup that I have.
And I'm guessing if you don't touch them, probably stick 7 to 10 days, right, before they fall off.
Yeah, but I have to change my sensor
Yeah,
every
no, you
4
have to
days.
change
So
every 4 days, but if you didn't, then 7 to 10 days. is
7 to
probably
10
good.
days. Is the thing. And after that, people want to change anyway because it's, think of it like a Gillette razor blade model. Like when you use a fresh blade, yeah, afterwards it gets start wear out and you throw it.
What's the sample rate of this thing? How
Good question. So today we started at 60 seconds. Then right now in our studies, we are running it at every 3 minutes, primarily because unlike glucose, these markers don't change that much.
You want to say?
And I'm saving more battery life. I'm saving longer durations.
And it's Bluetooth to the phone then, or?
Yes, standard. Somebody was asking me that. So I worked at Corvo, where I looked at new RF technology. I don't want to be testing new ones. I want to do tried and tested,
Yeah.
and I want low power and low cost. Because ultimately, it's a low.
Well, you could do FTTT as well, right? Or mosquito, or 33 megahertz. Bluetooth is, like you said, more test.
But then there is a IPA. There is one thing that I need to make sure is IPA security.
Lower power.
So does it store data if you're away from the Bluetooth sensor?
It stores in the device, and then we do a sync to pull it up. And I pull it up, it goes to my server, and I put it in my server where it's behind a firewall. So today, the sensor and the device just a means to an end. Ultimately,
Just out of curiosity, which Bluetooth chip you use?
NRF
That's what I was asking, NRF 52.
Oh, okay,
standard.
cool. No, it's standard, it's just one of the minis.
One of the many things.
It is, no, it is.
So I covered this. Yeah, okay, outside IBD, we also do stress. In stress, people ask us to do cortisol, which is one thing that we're looking at. And we are also working with, we're looking at traumatic brain injury, people with PTSD. We work with military cohorts. There's a lot of interest from the Army and the Navy to look at injury-related thing on a stress-related activity. Also, we are working with some clinical groups to look at people with psychosis, schizophrenia, multiple disorder, and so on. So cortisol is another area. Non-alcoholic fatty liver, it came as a interest by one of our clinical groups. And liver is an organ that repairs itself and also can be destroyed. Like if you abuse your liver, that's what gets to cirrhosis. But liver is the one that produces most of your proteins. And so that was our interest for partnering with them. But liver disease is something that is of interest for us. And we're also looking at rheumatoid arthritis and so on.
So in general, if you had other things that you wanted to measure, say lactic acid,
Yeah.
or something like the beer pheromone, or something.
Correct.
To develop some of the sensors to measure something, can you just talk about that cycle and what's involved?
Okay, these are all the markers that we did. In fact, I kind of skipped through it. So these are all the markers that we know are there in sweat. So there is lactates, there is glucose. We are funded by NIH to look at EPG, which is a marker for alcoholic addiction or alcohol deterrence. There's also amino acids, cortisol, cytokines, chemokines, neuropeptides, and there's also vitamins. Like GFAP is a marker for traumatic brain injury. A lot of these markers that you see are cytokines and chemokines, primarily because the ask came from BARDA. And I'm a small company. I can't put my resources. So wherever the funding comes and the ask comes is where I've worked in. So a lot of those came from there. We are doing melatonin for sleep monitoring, then cortisol, IL-1 beta. We also are doing glucose, but I stayed away from glucose is primarily, I don't want to compete with Abbott.
Yeah.
Okay, it's too big. And
And it's not painful, so whether it's an injection or not. I have the problem in the older version that you scanned. I've scanned this arm and, oh, it's on this arm. I didn't know which arm I put.
Yeah. So going back to your last questions, people ask me about vitamins. The challenge with vitamins is we can measure if it is there in sweat, but the next step here is building the threshold. For building the threshold, are you willing to pay for it to know what your vitamin levels is? Like
Where-
today, we
Which
did.
one has the most money? I mean, we get down to it, who has the most, where's the most money, pick your company and go somewhere. Which thing you measure that will generate the most revenue?
It's not what we measure, it's what use case we go after. Like IBD actually is a small population, but you actually have more return on investment. Because if you're cured with disease and there's nothing like any other solution, I'm the only one in the game. All I have to do is provide the quality to say I can benchmark it. But vitamins, there's no threshold. What is the acceptable level? So we're looking for, like, remember I showed you with the calprotectin? I know what an active state and a remission state is, what a disease state and a healthy state is. With vitamins, hey,
But, so most of your work, or perhaps all of your work, is the type of work you would have to get approval for. You talked about getting approval and having to do clinical studies.
yes, but not necessarily, because I'll take the case of Apple. When Apple put out their heart rate variability, they put a disclaimer saying it's not a medical device. Right. So what they did is they put it out, they started selling, people started using, the usage analytics came in. They now use that value to build and re-optimize. Then when they got their critical amount that is needed to pass FDA, they're actually putting a case for a 510K to get this certified as a medical device. So it's all how you plan.
Sure, absolutely.
Yeah.
But today you have
Sorry, go ahead.
lactic acid.
Yes.
Today you can do lactic acid.
Yeah, we have done one work, but we didn't proceed too much on it.
Right, because you need a funding. Got it. Okay, thank you.
Is there a marker for depression?
Yes, that's what I said. Multiple MDD is multiple depressive disorder. So I didn't put any slides on that. There has been interest from bipolar disorders and schizophrenia. We are starting to talk with the Dutch group. There is interest in those markers.
So I want to talk to you after. Any markers for cancers?
All the markers are for cancers. In fact, so what happens in a cancer is your inflammation, your body basically sends signals that it's going on an inflamed state. In a cancer, you are in a hyper-inflamed state because of the cells going, right? All of these, IL-6, TNF-alpha. So TNF-alpha has a half-life of 15 minutes. IL-6 has a half-life of 30 minutes. CRP has a half-life of 19 hours. So to me, monitoring CRP is like a big half-life. Blood test can do it. You can go and take a blood test every week and do that, and you can see it. But you can't monitor TNF-alpha. That's why I focus on TNF-alpha, which is a short half-life.
What about like illicit drugs or testosterone?
So we have done DHEA, and we did, so my partner, my co-founder, she does a lot of the research, R&D side. She has done THC, which is marijuana detection. So we have done neuropeptide Y, which is all about stress and so on.
THC, fentanyl.
Fentanyl, yes.
Well, on the THC, can you discern between the CBD type and the more?
You know that
This is
question.
This is the paper that we got published.
So law enforcement, I wanted to talk to you.
Yeah, we did. We presented at the Texas and Oklahoma Law Enforcement Conference.
Yeah.
A couple of years back.
That was my.
I have to pick where I put my chips in. It's like playing blackjack. I can't play so many hands. I pick the one that I want to. That's what. So yes, but our platform can do it. Yes.
So your basic sensor is on a semiconductor, right? So you're.
No, it's not.
It's not now.
Okay, another question? Okay. So
How close are you to being commercially viable?
I don't have that, but my plan here, so where we are right now is Crohn's and Colitis Foundation has invested. So this is a study that I'm running right now where I'm doing 1 month across 100, that's what I call MVP, minimum viable product, where I'm looking at 100 subjects of different type across different sites. And then plan to go to Q submission with FDA. At this, the data from this will finish towards end of Q1 of next year. plan to do a FDA Q submission and then do a full clinical trial. Clinical trial typically, if I take Abbott Libre as a predicate and do the same type of trial design, would take around 6 to 9 months to finish. So I'm looking around a year
You're
and a half.
looking at
from there.
2025.
But that's not stopping me from working and giving the device out, as long as they say it's not a medical device.
But as long as if you're doing things that are not medical, like drug detection, then you could go faster.
But FDA doesn't look at it that way. FDA looks at it by use case. So why FDA? You need FDA to get into insurance pay. Okay, like what Mike said, that CGM is darn expensive. Average person cannot afford it. But with insurance, they can afford it. You need the insurance copay to get the bigger population. Otherwise, it will be only for the people who can afford that platform.
What are you looking at price for? What would be the future production today without shut? What would be the price point?
It's a function of volume. So today, for IBD, we are looking at $200 per month, which is basically using the CGM as a model. But then, depending as the volume goes up, because we can even get it down to $50 per month for the sensors. The readers will be a one-time cost.
Need more subjects?
Yeah, always. Sorry, who asked?
Go ahead.
Okay, always.
Yeah, my sister has IBD and she's in Sweden.
Please sign up. There's ibdwearable.com. That's where we are looking. We're signing them up, and please have a sign.
So obviously your casing is all good 3D print, right?
For now, I'm doing 3D printing, but that's because of the volume. The reason is then I can do injection molding for that.
You go to a prototyping shop at some point. So, prototyping shop.
Thank you, salesman. With a commission?
So
Got
it's
So with-
10 minutes to 7. Yeah.
So with IBD specifically, usually the worst hit case is colostomies and stomas. And are you also testing post-colostomy in those patients that do have stomas and things as well? Because obviously that's a big sell to the insurance company, is to prevent radical surgeries. That's usually the extreme of
Yeah,
IBD
but
is where you wind up.
so the thing about IBD is the focus is more of, you can either get the surgery and then you get call it lobotomy or right? And then you have to wear a pouch.
Yeah,
which
so
is
we have a colostomy now.
correct. But then the whole reason here is to avoid doing the surgery.
No, absolutely. But are
So-
you doing pre and post? is what
Yeah,
I was
but-
getting at.
But we have not tracked the particular
Oh,
cohort.
yeah.
Primarily because we didn't see the value. Because once you get the surgery, people are saying, I have enough other complications.
Because even after post-op.
Yeah.
Most patients still have radical IBD problems.
Yeah, but
Because
they have,
they can't
but have
remove
not,
everything, right?
in my
Okay.
studies, I've not focused on that. I'm
Okay.
looking at more like people.
Because that's
without the
ultimately.
surgery. Yes, I know.
From the insurance, that's what they want to
That's what they
avoid,
want to avoid.
Because it's very costly healthcare post-surgery. Saying that as a former nurse, so.
No, that is very true. I don't have IBD, but over the last 2 years, I spent with the IBD community. I
It's very
feel the
expensive.
pain.
It's very expensive.
It is very painful disease. Some of them, it's very painful. And the fact that you have to carry a bag and you have to, it's,
Yeah.
People find it even disgusting to talk, but imagine if you are having the disease and not able to share,
The rate of
and that
infection.
creates a lot of mental stress, trauma, and so on. Okay, yes, go ahead.
Could you introduce a little bit about the algorithm that you use to analyze the data?
That's my secret sauce. It's like you're asking Abbott, tell me how your CGM algorithm is optimized. Abbott is not going to give it away. Yeah, but it's
Good
machine
try.
learning. Yeah.
Don't ever get anything you don't ask for.
Yes.
I do have a question related to that. Perhaps you don't want to answer this question either, but if, is it the computation for the algorithm, is it on the phone or is it in the cloud?
It's in the cloud.
Okay, so you put you do like an airplane, it's not gonna work, right?
Yes, so right now, because I'm in the clinical development stage, I do everything in the cloud, but eventually I will translate it down to the phone.
Is it like a significant computational load or is it
No.
like suitable for edge computing?
I don't know I didn't think about the last part, but it's not a significant computation.
So you're using for correlation, you're using time series database based on reads.
Yes, broadly.
That's your really.
It is not, because if the binding happens, the analytical signal is so strong, I don't need to rely on any more machine learning rely algorithms to make it final, like how you have to do with your heart rate and so on.
It's formulaic.
Because my analytical binding is very strong. That's what my core relies on. In fact, my sensor is relies on the binding. Which is what I told you about the capacitive.
I mean, you may want to think about whether you want to sell an side on the phone in terms of like, so I have pressing reverse and your back.
Absolutely. That is in the center of my focus right now. Yeah. Yeah. The reason I'm collecting all of this into the server because I'm looking at it, combining, and then looking at it across populations, across forms. But eventually, once we optimize the algorithm, we push it to a more user-friendly thing. Yes. Okay. I do want to, I know 10 minutes to the end. So there is another platform that I didn't talk about, which is called the REAP platform. And somebody asked me a question here before. If you can do 2 markers or 4 markers, can you do 12? Can you do 16? Can you do 20? And that question was asked by us by the BARDA. So we have this platform, which is basically the same electrochemical sensing, but translate into a PCB type of chip that I have. The electronics here is the same. I just made it bigger because I could put a bigger battery, but the whole point was to fit it in the palm of your hand. This ask came from BARDA because BARDA said, well, I like what you guys are doing, but I want to change the marker as I need them. Because today, COVID is due to one viral infection. Tomorrow, there could be a different pandemic due to a different virus. There is bacterial infection and so on, correct? So, I said, well, we could expand, we could easily scale up, and that's how this platform came about. And the second ask was, can you make it CLIA-waived so that people can test it at home? CLIA-waived is not to go to a clinic or not to go to a hospital and test it. And so this is where we said, well, you don't want blood, so we're looking at saliva, urine, and to go look at those markers in this. So it acts like a dipstick, or today we do it with a pipette on top, and it can customize to a dipstick. And where we are now working with them is to see if you can evaluate for the sepsis monitoring. Yes.
So you're looking for FDA approval for the US, but you can market it without FDA approval elsewhere.
That's right, because I'm funded by the military for this.
So is the Gates Foundation a target for you?
Gates Foundation, they like fancy stuff. They don't like practical stuff. So it's a waste of time for me. Yeah.
So you said that this would be available for sepsis.
Yeah. So we call this the READ. For the medical, it's a near patient handle point of care platform for inflammation monitoring and sepsis progression. Sorry. Sepsis is a complicated thing because you have like the arc of sepsis where you want to be able to predict the likelihood of a person dying. So the use case came from the military because the military said, if I have a makeshift hospital in like an Afghanistan type of base, like, and a person goes through surgery, the likelihood of that person developing sepsis is high because they're exposed to a lot of virus and bacteria. For them to, it's called a resource limited setting. And for them to decide whether this person is going to develop sepsis or complications of sepsis and has to be airlifted to the nearest NATO base, which is a big cost on the taxpayer income, or can the person's body fight back and cure itself with medication or with treatment? So to do that, that's called a 28-day mortality. That is what the signal is here. So they gave us a combination of markers that we are actually optimizing the platform to do, which will tell the risk score of a person developing sepsis due to complications.
And obviously, if you get sepsis once, you're more likely to get it again.
Yeah, that it goes back to how you see. That's a very subjective, because if you fall ill once, with the likelihood of falling ill again really depends on how one takes care of their body post recovery, right? You could treat yourself and keep it and avoid those complications. Yeah, but the susceptibility is high. But here we're looking at military cohorts and so on. That's why we're looking at whole blood, serum, plasma, urine slides.
So are they already using it now?
I'm testing with them. I'm in contract with the military. Then the non-medical use cases where we're looking at food pathogens and food pesticides and water-based like Salmonella, E. coli. Like today, you see E. coli or Salmonella that you get, like you bought something from Kroger and you said, I washed it and I found that is a Salmonella. You could just take the runoff from the water and you can test it. This is what Vikram's focus and PhD work has been. So we are starting to explore this. We can also look at alpha toxins, rotavirus, cryptosporidium, and so on. All of this is an R&D stage on the right. And then I'm going to leave with this thing, which is the One Health. This is what Ed told me to share. Vikram, Ed will be coming in 2 months from now, and he will expand and talk about all the One Health monitoring. So you want to talk about it, Vikram?
Sure.
Go ahead.
That's part of digital farming.
November 2nd will be the day.
So it's a little teaser that I'm going to leave Vikram to finish off my talk. So what he's going to present in 2 months.
Just giving a little bit of a gist about One Health. So the idea of One Health came when the other co-founder, Dr. Shalini Prasad, we were talking about it. We were like, okay, there's two sides to biosensing. There's one side with all the things that Dr. Muthukumar was explaining, where about post-diagnostics, where we were trying to look at the precursors or the triggers from it. One of the major precursors can be the soil itself, where most of the food is grown. So we wanted to look at a biosensor capable platform that we can actually deploy in soil and in food, in the body. So it's three things. So that's why it's human health, environmental health, and food health. So that's why it's the One Health framework. We tried to combine these three things. And we were trying to look at a precursor or environmental triggers to human living. So that was the broad idea behind it. And what gelled well is that's exactly what the UN said about the sustainable development goals. That those are the SDGs. There are 17 SDGs right now, which are put out there, which pretty much talk about how the soils contribute for a better production of food, which then translate to better nutrition, which then translate to a better environmental health as a whole, which then means better human living conditions. So that's the idea we took, and we built this One Health framework where we took soil as a core, and we looked at different things, including the soil physical and chemical parameters. We looked at soil nutrients, which are drivers of what the soil state is and how the farmer can either grow things or essentially not grow things. And also soil carbon, which is a major factor or a surrogate marker of how the soil health or environmental health, including climate change, can currently be diagnosed. And that's where the big players like Amazon, Nestle, all the big food and the industrial players come in, because they like soil carbon, because that's a way to evaluate carbon neutrality and incentivize their corporations. Exactly. So there's incentive on both sides, more on the industrial or the institutional basis, but also towards farmers. So if farmers have good regenerative practices that contribute well to that landscape and structure, then they get incentivized in tax rebates and so forth. That's what the EU is planning, not so much the US yet, but yeah.
Problem is nutrients in soil is
It's deadly. So currently the way it's done is you need to take a soil sample, send it to a lab, which is also expensive, but the soil is no more a representative sample because you're now transporting it, and there's a lot of things that goes on in soil. So essentially things change. That's right. And the platform that we developed was a as part of ecometrics, we actually developed a soil probe, which has multiple sensor slots that actually go into the soil and it measures over time.
So what we found out was we use electrochemical methods. We found out that gives a better accuracy.
Yeah.
Now, if you were to, what I presented about human health, right? If you were to now change that and say, if you were to treat soil like a human entity,
yeah,
and say, hey, illness to wellness, wellness to illness and back to wellness, and treat the soil as how can I preserve the soil health and make it more stronger, more regenerative, then the whole nutrient that goes from the soil into the food that we eat can also be enriched.
You
So that's
understand the
what-
farmers don't give a damn about that, right?
Yeah.
This is where we are participating with.
Actually, the Midwest states up north, right? So Midwest is already budgeting, the agricultural department start looking into this already. It's
So-
Yeah,
already-
they're looking into it, but the farmers themselves, first to buy a sensor.
If they are asked to,
Their number one thing is nitrogen. And they want to know.
exactly.
They want to know, are they deploying enough? Are they deploying too much? Because their crop is deploying too much. Are they doing it too early? Are they doing too late to the crop cycle? Too little, it doesn't impact the crop. If they do too much, then they get a runoff, and then they get an algae bloom, then they get an EPA fine. And if they do it too late or too early, they can burn out the crop or
Right.
some things. And nitrogen costs $200 an acre.
Exactly.
Not that I know that.
Sensors have.
Wonder how you know.
Nice guess.
Yeah, not only the calibration aspects, the accuracy between lab to lab. So
Oh, yeah.
like you mentioned, Midwest labs versus Texas labs versus California labs are offset.
So question for, so if you can control the whole food chain. you
Yeah.
know,
Right.
Growth to human to.
Right.
How much disease can you prevent in the population?
So the idea then becomes, that's what we call a holistic or a 360-degree monitoring. So you look at every aspect. So I'll just give you an example of what I saw and my personal basis on this. So I actually went to a conference. That was the World Soil Congress in Scotland last year. And one thing they mentioned there is, so when the conference is done, everybody goes for a lunch. So they actually served lunch there. And when you actually go to pick up your food, you have a few options. Next to that, they have like a placard there, which shows where everything was grown, how much pesticides was present in it, like a accuracy rate, and then what are the different nutrient levels that was grown in, what type of soil it was. They have the certificate or a card there that said, okay, it's a quinoa salad, where was the quinoa grown, right? Where was the kale grown? All of that. So that's kind of like a,
Yeah.
like a Bitcoin based.
It's a quality control system.
It's a quality control system.
And the other side of that is the pharmaceuticals are
Yeah.
going to give you a lot of
Yes,
feedback, right?
exactly.
They make money off of this.
Exactly. But it's a teaser. It's a teaser.
Yeah.
More about that.
Come back in November. Seconds.
Last question? Absolutely.
Thank you.